Hello, and welcome to today's webinar. I am Robin Brumble, a registered nurse, as well as CLL Society's Director of Scientific Affairs and Research. At CLL Society, we are dedicated to bringing credible and up-to-date information to the CLL and SLL community because we believe smart patients get smart care. As a reminder, you can rewatch all of our educational programs by going to the section of our website called Education on Demand. We would like to thank our following partners for supporting today's webinar. At this time, I would like to introduce our moderator, Dr. Brian Kaufman, who is CLL Society's Executive Vice President and Chief Medical Officer.
Thank you, Robin. I would like to welcome our audience and speaker to today's event. We're joined today by a great CLL, chronic lymphocytic leukemia, expert physician, Dr. Mazyar Shadman, who's a member of our expert medical council at the CLL Society and is also an associate professor at the University of Washington School of Medicine in the Division of Medical Oncology, and also an associate professor at the Fred Hutchinson Cancer Research Center in Seattle, Washington. I will also add that Dr. Shadman was one of my personal physicians taking care of me when I had my CAR T therapy up at the Seattle Cancer Care Alliance. This is a particularly important topic for CLL patients who have relapsed or refractory disease. And I'm an example of those patients. I participated in the clinical trial to receive CAR T therapy in 2018, and has provided me with years of remission. The CLL Society has developed a lot of helpful information and resources around this therapy, which can be found on the resources section of this event and on our website under CLL Information and Management and Treatment and Research. One new resource is our CAR T therapy brochure, which helps explain this therapy to patients. You can view this information online, or you can order a free paper copy. We're excited to welcome Dr. Shadman, who will be sharing much more in-depth information about CAR T therapy and some other cellular immunotherapy options, and we'll have lots of time to answer your questions. Thank you, Dr. Shadman.
Hi, my name is Mazyar Shadman. I'm an associate professor of medicine from Fred Hutchinson University of Washington in Seattle, Washington. It is my pleasure today to talk about CAR T therapy for chronic lymphocytic leukemia. And I would like to thank the CLL Society for the invitation and for the opportunity of having a direct interaction with our patients and caregivers. These are my financial disclosures. As a clinical investigator, I work closely with companies that make the drugs that have made a huge difference in how we treat chronic lymphocytic leukemia and lymphoid malignancies. So the focus today is on CAR T therapy for CLL. You may have seen the excellent news earlier this year of very long-term remissions of patients with leukemia, actually chronic lymphocytic leukemia or CLL, from CAR T therapy. And the example of two patients who achieved more than 10 years of remission after treatment was all over the news and caused a lot of excitement, really indicating the importance of this very important treatment modality for lymphoid malignancies in general, and for CLL as one of the first diseases that this treatment approach was used for. And these are patients who were treated at the University of Pennsylvania. And there was an important report in the medical literature, followed by coverage in the media about the long-term remissions from CAR T therapy. So I think it is very important for us in the CLL community to talk about this treatment approach. And for today, we have a few topics that we would like to cover, starting by what are CAR T cells? So we'll talk about that. Why do we need them for CLL? We have so many treatment options. Do we really need another treatment modality? Then a little bit more specifics about CAR Ts. How do we make them? If you're using them in CLL, when would be the right time and what's the right setting for using CAR Ts in CLL? Does the treatment comes with a risk? What are those risks? It's always important to understand the risks and benefits from any type of treatment. So we'll cover that. And then what's going on in the field and what are the latest changes, what is going on in terms of clinical trials and for CAR T specifically, and some other immunotherapy modalities that we're increasingly utilizing for treatment of CLL as a disease that's considered to be a lymphoid malignancy or a B-cell non-Hodgkin lymphoma. And then we'll conclude with a few take-home points. So what are CAR T cells? If you think about T cells, and the example that I always use is using T cells as a soldier or as soldiers that protect us, mainly from infection. So these are very capable and very strong immune cells, the T cells are, that are just not trained and are not optimized to fight cancer, in this case, CLL. And by the way, when I use CLL, it refers to both CLL and SLL. So I just use the word CLL, but everything that I'm mentioning today applies to both CLL and SLL. So what happens in this treatment, we basically train and we teach the T cells of each patient. So they recognize and they attack the CLL cells the way they attack infection etiologies, for example. So really, this is a fancy word for training the T cells to be able to attack the cancer cells, in this case, CLL cells. In fact, CAR T therapy, which stands for chimeric antigen receptor, and I have a cartoon in the next slide and I'll explain what that receptor or what that CAR is. The CAR T therapy as a treatment approach or as a treatment modality has been a major advancement for treatment of different types of lymphoid malignancies. In fact, the CAR T approach is currently approved by the FDA for the examples that you see there: diffuse large B-cell lymphoma, follicular lymphoma, mantle cell lymphoma. These are some of the non-Hodgkin lymphomas like CLL. We also have approved CAR Ts for acute lymphoblastic leukemia, a form of acute leukemia, and more recently with multiple myeloma, which is another type of blood cancer. It's important to note that for CLL, and at the moment, in August 2022, CAR T remains to be an investigational treatment, meaning that it's not approved by FDA for physicians to just prescribe it and use it. So if a patient is interested or is eligible, or if CAR T is the right treatment for a patient, it has to be given as part of a clinical trial. So patients need to sign a consent and participate in a clinical trial in order for them to be able to get access to this treatment. Now, let's look at the CAR T and kind of be a little bit more specific. So on the top panel, you see the before. This is the situation that our T cells have. The big orange cell there is a T cell. This is an immune cell. This is a soldier. It's a fighter. And the blue cell is a CLL cell. You see on the surface of the CLL cell a few proteins. Basically, each protein has a name. You see the ROR1, CD19, CD20. These are just a few examples of different proteins that are expressed or are present on the surface of the CLL cells. So this T cell here, the top panel, is not able to interact with this cancer cell. And that interaction is needed for the T cell to kill the cancer cell. So cancer cells kind of escape from the T cells because there's no way for the T cell to get engaged or to get attached to the cancer cell. Now, the after part shows you this black piece of molecule that is that chimeric antigen receptor. This is something that we make in the lab. And in a process that I will show you shortly, we'll put this molecule on the surface of the T cell, and that changes the name to a CAR T cell because it has the CAR receptor now added to the T cell. What we do by this procedure, we enable the T cell now to interact with one of the proteins on the surface of the CLL cell. In this case, I'm giving you the example of CD19 receptor because The CD19 targeting CAR T cells are the most advanced ones in the clinical trials. And the CD19 directed CAR is the CAR that will most likely be approved for treatment of CLL at some point, we expect. So as you see here, we basically what we did, a T cell that was kind of not interacting with the CLL cell is now able to interact, make the connection. And the good news is that the T cell's job is to kill the cell that is enemy. So right now, after doing this, CAR T is able to create an immune storm. You know, basically the graphics that I put there mean that there is an immune storm, there is an interaction, there is a lot of proteins, there are a lot of proteins that are made as because of this interaction. And the result is the cancer killing. So these proteins, and by activating different pathways, this CAR T cell is now able to kill the CLL cell. So of course, a very simple cartoon here, but just the idea was to show you that CAR is basically a synthetic protein, a piece of protein that we make, and we somehow, by the process of genetic engineering, we add it to the T cells, and in a process that I will show you shortly, we make more of these CAR T cells. And when these CAR T cells go back to the patient's body, they kill the CLL cells in this case, or lymphomas, different type of lymphomas or leukemias that I showed you in the prior slide. So in principle, it's just training the T cells and giving them a way to recognize the cancer cell. Now, remember, CAR T is not a specific name of a specific drug. It's an approach. It's a treatment modality, meaning that we can have different types of CAR T products, even for one disease, including CLL. So for example, I showed you different proteins, like I showed you in panel A, the CD19 as a target. We could do the same thing with CD20, which is another protein on the surface of cancer cell. Or we can pick another protein called ROR1. And these are actually real examples. There are CAR T products that are being tested in various clinical trials targeting either CD19 or 20 or ROR1. So really the point is that this is not just one drug. This is opening a door for a new modality, a category of drugs that could be available, and that could each be different in terms of what protein on the cancer cells they target. And you can imagine how many combinations and sequences, and so there are many treatment options down the road in the pipeline currently that are being tested, and hopefully we will have access to them as they get approved in the next few years. This basically is a more technical picture or a graphic of what I showed you. It basically shows that when the T cells are in the top T cells are in the blood vessel, then they get out and they attack the tumor cell. The tumor cells die, and what's inside the tumor cells gets released to the blood. And that activates the immune system. So it's not just the direct attack of CAR T to the CLL cells. This process can activate the immune cell against the cancer cell, which is a very amazingly interesting scientific advancement that we've had in the past 10 years, as I mentioned. Now, why do we need them for CLL? We have so many great drugs. We have good drugs, so we all know that, and I tried to summarize them here. But we still do need treatment options for patients with relapsed disease. Our current options include BTK inhibitors. These are a family of drugs that target this enzyme called BTK, and we have a few examples: ibrutinib, acalabrutinib, soon zanubrutinib. We are expecting to have zanubrutinib also used. But remember, once ibrutinib or acala are not effective, we can't really switch between these three drugs. It's a different story if the patients don't tolerate one of the three. We can, of course, use other ones. But if somebody's disease is progressing on either Zanu or acala or ibrutinib, you can't switch between the class. So technically, these three drugs are considered one treatment option. So I'm just trying to tell you that we do need better drugs. We will have pirtobrutinib or Loxo 305. That's another drug that is not yet FDA approved, but we're hoping to see it. We have venetoclax. We have another class of drugs called PI3 kinase inhibitors. There is a lot of discussion, as you may have seen on the CLL Society website, about what will be the future of this class because of some concerns about infections. And there are monoclonal antibodies and chemotherapy, but the list is, there are many number of drugs that are effective and very helpful, but once you get past the BTK inhibitors and venetoclax, we really do need treatments that are effective because we do see patients who are in need of treatment. So we do need a treatment like CAR-T. I would also look at it in a different way. Right now, we need them as a treatment for relapse setting. But imagine a time that CAR-Ts are very effective and also very, very safe. And I will show you in a second that this is a one-time treatment. So CAR-T therapy is a one-time treatment. And for patients who have a lasting response and persistent remission from it, you can imagine getting a treatment for one time, which is a lot of work. It requires a couple of months of really hard work for patients and physicians and the medical team. But then you get years of remission from a one-time treatment. So I think there are people, including myself, that believe that if CAR-T becomes optimized, meaning that it's effective and it's also safe, it could potentially be used in earlier lines of therapy, maybe even in the first line or second line. And that way we can provide a long-lasting, treatment-free and disease-free remission for patients. We are not using the word cure, but at some point, with all the great drugs that we have, and also modalities like CAR-T, we can think about combining these, and long-term remission at some point will be a cure. So that's the goal. We're not there yet, but we do need CAR-T for these two reasons. We need them today for patients with disease that's active after using other treatments, and we need CAR-T as a potential treatment modality for even early lines of therapy, including first line. Now, let's walk you through what CAR-T process is and what it means for patients. And I will spend some time on this slide, and I'll walk you through the process. And I will explain at the same time what it means for the patient logistics. So here's a patient, and we'll be following the numbers here from 1 to 5. So when we have a patient here on the immunotherapy program, for example, we start by doing an initial assessment, making sure that patients are in a reasonable shape in terms of other medical issues, and as long as they're eligible. And as I mentioned, these days you can only get CAR-T as part of a clinical trial, so patients need to be eligible for that specific protocol. But then the next step, once they're eligible, is to do this process called leukapheresis. What it means, it basically means that we take their T cells out of their body. And this is a process that takes a few hours. It's an outpatient process. There's no need for hospitalization for it. And basically all we do, it's a process very similar to dialysis, when patients will be attached to a machine and the blood goes through the machine and comes back, and we just get the T cells or the product that we are interested in. And then this product will be sent to the lab that is responsible for modifying those T cells. That genetic engineering process happens in the lab. Now, in the meantime, as you see here, number 2, this is all the fancy work that happens in the lab, which usually takes 2 or 3 weeks. So patients during that time, they could be very stable. If their disease is stable and under control, we send them back home sometimes. Sometimes they stay with us, but we don't really need to do much. But some patients have more unstable disease, and we may need to provide them with treatments in the meantime while we're waiting for their T cells to be made. And as you see on number 3, finally, they make these modified T cells and they expand them in the lab, so they get reasonable number of them. And at some point, number 4, or before number 4, these T cells, now called CAR T cells, you see the shape of the T cells have changed because now that they have that receptor or that protein on their surface. Now, before we reinfuse those T cells to the patient, we give them some chemotherapy. So that chemotherapy is a chemotherapy that's given in the outpatient setting in the clinic for 3 days. And really the intention is not to treat the CLL. The chemotherapy is given to make room in patients' immune system for the new T cells to go in and to expand and to make more of themselves. But this chemotherapy is important and it's necessary. And the two drugs that we use are number one, fludarabine. Number two is cyclophosphamide. So FC from the FCR regimen for those of you who are familiar with that chemotherapy. But this would be only one cycle of the two drugs. And we usually give them for 3 days, and there will be a couple of days of no treatment. And then patients will receive their modified T cells reinfused back to them. And that would be the time of, basically, we call it the day of treatment. And really what happens after that infusion is basically a lot of monitoring that we need to do. What happens is that with these T cells now being able to attack the CLL cells, they do the attack by creating an immune storm in patient's body. So that's how the immune system works. That's how the immune system kills. The symptoms could be very similar to having a virus, a viral infection like flu or maybe COVID, when patients may get fever, some general achiness, muscle pain, joint pain. More severe cases could be changes in the oxygen level or blood pressure level. In CAR T in general, I'm not referring to CAR T for CLL. In general, CAR T therapy could be associated with a drop in the blood pressure or oxygen. So sometimes patients need to be monitored very closely in the ICU. Almost always this is temporary, and we just need to be able to support the patient to kind of go through that immune storm. And there are other side effects, including some confusion and neurological toxicities that usually happen in the first couple of weeks. They can be in different severity grades, sometimes very subtle, sometimes more prominent. And we do have very effective treatments these days to control the symptoms. And as I said, they're almost universally reversible, but it requires a lot of supportive care. So we usually keep our patients very close to us for 30 days after this point of CAR T infusion. If you see number 5 on this picture, the process of T cell manufacturing ends there and the infusion happens, but the clinical monitoring starts. And we watch our patients for 30 days. It could be in the clinic and seeing them in the clinic. It doesn't mean that they have to be in the hospital. If they get any of the side effects that I mentioned, of course, we will monitor them in the hospital. But there are many patients for CLL specifically who don't spend the day in the hospital. So it would be a very close monitoring in the clinic, but not a regular clinic. This is a very intensive clinic where we do a lot of close follow-ups and clinic visits and lab draws and things like that. So this gives you hopefully a very big picture understanding of how the process is like. As I mentioned, it's a one-time treatment, but it's kind of a pretty involved one-time treatment that patients usually spend around 7 to 8 weeks with us. And the first few weeks are related to just being ready and being tested and making sure they're in a good health and getting their T cells out. And the second month starts from the day of their T cell infusion and the close monitoring that we do to make sure we're capturing any side effects and we're treating them appropriately. Now, when should we use CAR T in CLL with all the great drugs? I would just make 2 points. It's, in our experience, it's very difficult to manage patients before CAR T if they come to us too late, meaning that if they have tried all the treatments and there is not a reliable and effective treatment that's keeping their disease under control, and then they consider CAR T, we usually have a difficult time doing the process that I showed you and controlling their disease at the same time. So it's doable, we do it. Sometimes we're not successful because it could be a challenging situation. So as somebody who's heavily involved and takes care of patients with high-risk CLL needing CAR T therapy and similar approaches, it's extremely important to think about a treatment like CAR T early on when your disease is still responsive to treatment, when the disease is stable. So that's critical. And, it's also kind of another way of saying that. And as you see there in the 2 points that I made on this slide, always keep a reliable treatment as your backup plan when you are considering something like CAR T therapy. What does it mean? For example, if venetoclax is still working, but that's pretty much the last reliable treatment, the time of considering something like CAR T is not when venetoclax stops working. It's actually when venetoclax is still working. So you know that if for whatever reason CAR T doesn't work, which is possible. I mean, the success rate with these treatments are not 100%. We're at the 50% range. So you do want to know that you have a treatment that you can go back on immediately. So the timing is critical, and I can't emphasize on that enough. And these slides will be shared with you, so I don't want to spend time. This is basically a summary of what I just told you on the paper we recently published. It really talks about the timing of referral. This is for physicians, but I think as patients, we all need to make sure that patients know that the timing is critical, and the timing is when the disease is under control. So think ahead of time and have a backup plan when you're considering CAR T. I covered the side effects that can happen after CAR T, and this slide basically summarizes. When you have an immune storm, pretty much any organ could be affected, but I summarize the ones that are more relevant: the oxygen level, the blood pressure, confusion, but the medical monitoring would be at its highest for the 4 weeks after CAR T infusion. So the medical team will be watching very closely every organ, and the treatments are given if needed. My intention was not to share clinical data. As I mentioned, the product that may receive an FDA approval in future, and I have no specific information other than knowing that this CAR T product that has a long name, Lysocaptogen Marolocil or Lysocell or Beryanzi, has finished this study, the clinical trial, that will be submitted or is submitted at some point to the FDA, and the FDA may consider approval of this product for patients with relapsed refractory CLL, which will be a big change for our field and for our patients. You see the response rates of around 80% and a complete response rate of 45%. And this is just a phase 1 portion of the study that's published. We don't have the results of the actual the combination study with ibrutinib and the phase 2 piece or part. But I just wanted to give you the awareness that if we get a CAR T approved in the next year or two, this will be the CAR T, the product that goes for CD19 as the protein of interest on the CLL cells. And, it of course comes with a toxicity. The numbers are listed there: significant immune storm or cytokine release syndrome in 9% of patients, and neurological side effects, confusion, sometimes changes in the mental status, and issues with finding words and things like that. Grade 3 or 4 usually refers to more significant ones, and that happens in 20%, 22%. So these numbers are there, but again, we have gotten much better in how we manage these side effects and how soon we intervene. So, for patients who need them, these are manageable, and I think for the CAR T standards are acceptable numbers. And just a very quick point that there are studies showing that we can actually continue ibrutinib or drugs like ibrutinib with CAR T, and this is important. We may, in fact, get the CAR T approved in combination with ibrutinib. We'll see. But it is important for us to know that at least not only ibrutinib or drugs in that family probably are not harming CAR Ts. They may actually help the CAR T in terms of both working, and there may be a signal that the side effects of CAR T therapy may be less severe when you use a concurrent drug like ibrutinib. Of course, it has to be in the appropriate setting. And very quickly here, there are many studies that are basically taking advantage of our immune system and immune cells and train them in different ways to attack CLL cells. I'm not going to read these for you, but just know that we are T cells are one type of immune cell. There are NK cells. There are studies that are trying to use NK cells to train them to attack CLL. I mentioned that in this approach that I spend most of the time on, we use patients' own T cells. Imagine we can use somebody else's T cells. And so maybe that's even a better idea, maybe because those T cells are probably healthier if you use a healthy donor. We also don't have to worry about the whole process of getting the T cells out and sending them to the lab. We can much faster, we can get somebody else's T cells. So these are all ongoing studies, and depending on how, what research institution or center you have access to, you may have different options looking at different types of immune therapy cells. Just for the last 2 minutes, I mentioned another immunotherapy modality that's getting more and more attention. Remember, with the CAR T therapy, we basically put a molecule on the T cell so it would attach to the CLL cell. Here, the black colored molecule physically brings T cells to the CLL cell. So this is not a form of T cell therapy. This is a drug. This is an infusion or an injection that patients get for, and they're different drugs, so they have different schedules every 3 weeks or every 2 weeks or every 4 weeks. And what the drug does, it basically physically brings the T cells to the CLL cells. So it's another form of really engaging the T cells and immunotherapy, but it's very different from the CAR T approach, which requires that 2 months stay and getting the T cells out and changing them and putting them back. So just giving you an example of a very novel class of drugs that are extremely effective for lymphomas, we know that. And for CLL, studies are just becoming more mature. And again, this is another treatment that's not approved for CLL, and patients can have access to it in clinical trials. And I will just, the CD20 CAR is something that we are involved here at Fred Hutch, and I just use it as an example. But there are many CAR T products that are being investigated in different institutions. I just wanted to, as an active study that's going on in multiple sites in the country, in the US that are listed on the right side, are currently involved in this study. I just wanted to kind of mention that as an example of a CAR T trial. So for future, I think, most of us hope that a CAR T will be approved for CLL. There are a number of studies, and we're hoping to not only see CAR T coming for patients who need it in the relapse setting. At some point, hopefully, they will be effective and safe, and we can use them in first line. And I think of CAR T as an effective treatment strategy. Consider clinical trials. Right now, that's the only way of actually having access to CAR T. And remember, one last time, the timing is not when there's no other options and disease is out of control. The timing is when the disease is under control, but there are limited options. But keep a safe and reliable treatment option as your backup plan in your back pocket, because there is a chance that CAR T doesn't work, and we need to control the disease while patients are waiting for the whole process. So, and remember, CAR T is not a drug; it's a modality, and there could be potentially many CAR T products for patients in future, which is great. It's always great to have more options. Thank you very much for your attention, and I'm happy to answer any questions. And these are my contact info in case there is any specific question that I can help with. Thank you.
Dr. Shadman, thanks so much for that. I've heard a lot of talks on CAR T, and that was one of the most practical kind of nuts and bolts. How do I do this? What's the state of the art now? Where is it going? Where does it fit in? I deeply appreciate you talking about that and about the other possible cellular therapies and immunotherapies, bringing up the bispecifics. We have a ton of questions, and they keep coming in. And we're going to try to get to all of them. If we don't get to your questions, we'll give you an email afterwards that you can email to us at Ask the Expert. And that way we'll try to answer those questions. So let me start with a couple things that were asked. Tell us a little bit about who is a good candidate for CAR T in terms of their prognostic markers? We had a ton of questions. Is this going to be for 17p only, or will it be for any patients? Does it work better with some patients? What about IGVH mutation status? Where a lot of patients asked about their particular thing, 11q, 6q, trisomy 12. Can you address that as an issue? Is that a factor that you consider when looking at CAR T therapy?
Great question. First of all, thank you, Dr. Kaufman, for the opportunity. I try to talk fast because I hope to answer as many questions as we can. So let me start by using the example of moving from chemotherapy to all the novel drugs that we have today. There was a time that, for example, 11q or IGHV mutational status were extremely important because all we had was chemotherapy. Then we learned over time that if, for example, we use ibrutinib or drugs in that category, maybe those risk factors are not as important. And as the data becomes mature and we get more information, we've learned that, well, really ibrutinib, acala, they work for patients who have mutated or unmutated IGHV or they have 11q. So the point I'm trying to make is that some of these markers are relevant in the setting of the treatment that we're using. So what's important when you use chemotherapy or novel drugs is not necessarily important in CAR T. I'm not saying that they're not. The information we have today is very limited. The good news is the response that I showed you and the data that I briefly presented in one slide, these patients are all very high-risk patients. The patients who responded, the numbers we see with 80% responding, 40 to 50% having a complete response, a good percentage having no detectable disease. These were by definition, they had to be exposed to ibrutinib. A very large percentage actually had progression after ibrutinib. 65% also saw venetoclax. They had very high risk features, including complex karyotype and 17p deletion. And I think more than half had IGHV mutation, unmutated IGHV. And yet these are the results. So what I can say right now is that we don't think that any of those markers are predictive of a better or worse response. This is a completely novel treatment modality. It may or may not have markers that would explain its response. I'm sure 10 years from now or 5 years from now, we may have some understanding. At the moment, the way we define who's a good candidate for CAR T is mainly by their response to the prior treatment more than the molecular factors. What does that mean? If I have a patient in front of me who has tried, let's say, acalabrutinib for 7-8 years and the drug stops working, and maybe they tried another agent or chemo before that, and now they're on venetoclax, and the drug is working, they still have some disease. This would be a patient that I talk about CAR T very seriously, because I think it's more important to see what options you have in front of you than what you had in the past. So when I think about a patient today who've seen BTK inhibitors like acalabrutinib, ibrutinib, and they're taking venetoclax, I start thinking, okay, what's next? And if I don't have a great drug in my hand, I start thinking about a treatment like CAR T. Now, this can change. We have more drug coming in the pipeline. Pirtobrutinib is coming, hopefully more. And this thought process can change, but it will be an ongoing discussion and kind of reassessment of your options in front of you versus what's available in terms of cellular therapy. Sorry, long answer, but I hope I answered a few questions there.
No, very helpful. But let me push a little bit on this, because people asked about, will CAR T ever be frontline? And I've talked a lot about seeing CLL as a chess game, and you have to think not one move ahead, but 2 moves ahead. But as you pointed out, practically that's difficult. And one thing as a patient you're looking at is, if I'm doing well on this drug, why do I want to do a therapy that has significant toxicity, even in the best cases? So help us, one, do you ever see where CAR T might be used as a frontline therapy? Because when these therapies begin, as you point out, they treat the worst of the worst, but wouldn't they do better if they treated someone who hadn't been beaten up getting to you for CAR T therapy? Or do we see them as a second line therapy? It's kind of hard sometimes when there's only so few classes of drugs to say, well, I better keep something in reserve because I need to keep CAR T in reserve. So now I'm keeping two things in reserve. It gets difficult as a patient and as the physician managing that patient to navigate this. So help us understand that and where you see CAR T fitting in the future as it moves maybe up the line in terms of therapies.
Great question. And, as you know, we don't have the answer to most of the, you asked a few questions in one. So when we get CAR T approved, I hope in near future, we will be using it not in the first or second line. We'll be using it in patients who are high risk by the whatever definition that we use, which is going to be a combination of their molecular features and, more importantly, their treatment history. And that's the specific CAR T that's coming, and I showed you the toxicity numbers. But we have one goal in CLL, and that's curing CLL with the shortest possible treatment with fewer possible side effects, right? So what I was referring to is that if you think of CAR T as a modality and not a specific drug, we could potentially in 10 years from now we could have like 5 or 6 CAR T drugs for CLL. And imagine that there will be a CAR T that has minimal toxicity, great, and let's say 60% of patients can have that one-time treatment. Yes, that one time is 2 months, but if you think about it as one-time treatment in 10 years, it may be acceptable. Would I consider that kind of a treatment in the first or second line? I think so, right? I mean, let's put a label on or hide the name of CAR T and say we have a pill that you take for 2 months, and the side effect is this, let's say 5%, and you get 8 years out of it with no treatment and no relapse. That's a very attractive drug, if you don't put the CAR T name on it. That's how I see it, and that's how the CAR T field. It's a very young treatment. It will get better. It will get better. It will get stronger. It will get safer. And I do think that when we talk about time-limited therapy, which is currently basically a combination of venetoclax with something else, CAR T could be a main backbone for a time-limited therapy. Are we there yet now? And the second point is that our threshold for accepting side effects as physicians and as patients is very different when we're looking at a situation where there's no other treatment options versus first-line treatment when you have so many amazing drugs. So if a patient is willing to accept a 20% chance of significant side effects in the fourth line, they will not accept that 20% in the first line. So it's a really dynamic decision-making process. And one word I would say is giving yourself enough time to think about it and educate yourself. And that's why that early discussions are very important. These are difficult decisions to make when you only have 2 weeks to decide. But if you have 2 years, you will be prepared. And a lot of our patients need to plan for this. They need caregivers. They're coming from different cities. They need to tell their family what will happen in that 2 months. And they have kids and dogs. I mean, so these things need planning. And that's why early discussions and thinking about it will definitely help with the process.
We had a lot of questions on the FDA and where it's at with the FDA. And I know that you don't have any inside information that you share with us, but we see CAR Ts are available in other blood cancers. You pointed that out, but it seems slower to get over the finish line in CLL. Is there any sense of this? When it's projected and why it's an issue. Right now, it's only available in clinical trials, and they can be quite onerous, the clinical trials for patients. They're much more involved, often the monitoring and the commitment for the patient.
Right. So as you mentioned, I don't, of course, have any information from either the company or FDA or the process. What I can share is that the study that I has finished enrollment is probably going to be submitted to the FDA, and the FDA will look at it, and if they find it reasonable, they may give it an approval or not. So now, I know it's a very vague statement, but at least based on the study that's already done, it is possible that we get an approval. So best case scenario, I mean, in the next year or so, we may hear something. I think it's possible, but we have to wait for the announcements from either the company that makes this car or the FDA itself. But just knowing that the study that is already finished enrollment the data will be submitted to the FDA. I think it's good news. It means that we don't necessarily have to wait for another study to be done or finished. And then we have to see what the approval, if there is an approval, what label the product will get. I mean, will they limit it to a specific patient population after certain drugs, or it will be more open? So why is it behind other lymphomas? Part of it is, of course, CLL is a very different disease, as we all know, and I don't need to kind of talk about it. I know that you have a lot of information about that on the website, but also about what else has been going on in the CLL field. We had so many great drugs that see what happened in the past 10 years. So the progress and the advancement that has happened for non-CAR T treatments in CLL maybe was one of the other reasons that CAR T was not as moving as fast as other lymphomas, where they didn't have the luxury of having all these great drugs. So it's a combination of CLL being a little bit trickier, and different CAR T products were tested, and finally one is showing some more promising results, but also the field was not feeling the need that much because there were so many great drugs already available.
Yeah, we are a victim of our own success to some level. There's some sophisticated questions here about your immune status after the CAR T. Your CAR Ts are directed not just at the cancer cells, but all of the B cells that make antibodies. Do people need to get immune globulin IV or subcutaneously for the rest of their life afterwards? If the CAR T is successful, does the immune system reconstitute? Because obviously that's a big issue in the pandemic, is you're quite vulnerable if you don't make antibodies.
All great questions. So I'll try to answer one by one. Number one, yes, immune system will be compromised for many reasons. Number one, remember, we use 3 days of chemotherapy, and that chemotherapy is FC of FCR regimen. It's only one cycle, and it doesn't have rituximab in it, but both fludarabine, which is the F, and cyclophosphamide, which is the C, they're just chemo drugs that suppress your immune system. More importantly, we go for, let's use the example of CD19 CARs, because again, that's the most advanced one. CD19 is a protein that is on the surface of cancer cells, but it is also on the surface of normal B cells. So when CAR T works, as a side effect, your normal B cells also go away, which is not a good thing. I mean, that's why immune system is low. And IVIG could be an option for patients who need
That's
it.
intravenous immune globulin. It's a replacement therapy. Yeah.
Exactly. That's intravenous immunoglobulin. We don't recommend it routinely to everyone, but if we see that some patients are having infections and complications because of not having enough B cells in their blood, then the intravenous immunoglobulin, which is the product of those B cells, we can use it in different frequencies to kind of have an antibody on board to protect them against some infections. For COVID, for example, we recommend in our institution, and there could be differences between institutions for their guidelines. Here, number one, we give all our patients Evusheld before starting the CAR T process, so they're covered for that few months of post-CAR. And regardless of having vaccinations, COVID vaccinations in the past, 3 months after CAR T, we recommend that they start all over again with the whole set of their vaccination. So we do our best. Yes, there are risks, and we keep patients on prophylactic antibiotics like what we do after stem cell transplant. They will take an antiviral drug for a year and some other antibiotic prophylactic drugs for 6 months, and they will be closely monitored. So the long-term immunosuppression is one of the side effects that patients need to be aware of.
I remember getting a letter saying that I had to update even some of my childhood immunizations post-CAR T because there wasn't clarity about whether I would still be protected against those, and I had to go through a whole revaccination. But I think it's worth it. I do like, Dr. Siddiqui, who's another CAR T expert, has said, give me a month or two of your life, and I can give you this deep and durable remission. And I like the way you talked about that. But let's touch on the chemo, because that holds a lot of people back. And it's, we know that CLL patients are higher risk for second cancers in the blood and stuff. And we know that chemo works by just disturbing the DNA. That gives us pause. It's immunosuppressive. It can damage the bone marrow. Do you see a time where we may be able to skip the chemo? Is the chemo a critical part of the CAR T process? Is there a way around it?
Good question. Yeah, so yes, we do need the chemo, and the reason we need it, remember that the number of T cells that we make in the lab so we take the T cells, the normal or original T cells of patients out of their body, we send them to the lab, we work on them for a couple of weeks, and then we make a number of T cells, CAR T cells. This will be a very small number. If we will infuse it back to the patient through their vein, if the number of CAR T cells doesn't increase immediately or in the next few days or a couple of weeks, they will have no activity. They won't work. The number is basically too small. So the main expansion, or the number of CAR T goes up quickly in the body. So we really need that. The success is dependent on how good these CAR T cell, what we call is expand in the body. They make more of themselves. If we don't use chemotherapy, we know that expansion will either won't happen or will happen very suboptimally. So right now, and we have tried different versions of the chemotherapy in some, including ourselves, we tried to, for example, drop fludarabine from our chemotherapy at some point and only use cyclophosphamide for the exact same concerns that you brought up. At this point, the combination of fludarabine and cyclophosphamide seems to be necessary for our success. Some other CAR T products, not for CLL, have used other drugs. So in theory, it's possible that we could use drugs other than fludarabine and cyclophosphamide. And of course, we are aware of chemotherapy. But remember, this would be only 3 days of chemotherapy, which is like one cycle of any other chemotherapy regimen that we used to use. And usually you give chemotherapy in 6 to 8 cycles. And if you think about it, this would be a very short treatment. We would love to avoid it. We would love to have a way of not using it, but we've got to look at the big picture. And if you're looking for success at the moment, that is the way to go. In the CAR T world in general, yes, and we with other immune cells, including NK, and there is a lot of interest in maybe getting away from chemotherapy and using some novel drugs. They're pretty investigational at this point. So we're kind of stuck with the 3 days of chemo for now.
Here's a common question, and one pertinent to myself. Is there an age limit on CAR T? Is there a time where you say you're too old to get CAR T?
Great question. I keep repeating this. There is a CAR T and there is a CAR T. CAR T is a general name. So for some CAR T products that we use, we kind of start feeling a little bit uncomfortable using it for patients after a certain age. The one that will be approved, I hope, for CLL seems to be one of the better tolerated ones. And we have not had, let me remind everyone that the exact CAR T that will hopefully be approved for CLL is already approved for another type of lymphoma called diffuse large B-cell lymphoma. So we have experience using it. Side effects are not necessarily the same, but for large cell lymphoma, we are not using an age limit because the CAR T seems to be reasonably well tolerated. I can tell you on our CD20 CAR study, we have treated at least 2 patients older than 80, and they did really well. So it depends on what CAR you use and what's the safety profile. I think I don't recall kind of telling a patient that they're not eligible for age. Age is a number. If we look at the medical picture in general and medical comorbidities, but age by itself should not be a reason for this type of CAR T to exclude a patient.
Got to finish up in a minute or two, but you touched on a couple things that I'd like you to just tease out, if you can, really briefly. That's can people make their own T cells? Would that avoid the chemo? Or like, if you could go over the CAR NK cells, the difference between those, autologous versus allogeneic. Can you explain what those words mean and how you see off-the-shelf CAR T? We talked about that, and then they seem to have disappeared. Can you talk a little bit about that stuff?
Right. So very quickly. So in this process, which is the most mature one, and this will be the approval if you get one, using patients' own T cells. So you have to spend time getting it out, changing it, taking care of the patient, and then you reinfuse the T cells back to the patient. Now, there's a lot of interest in not doing that and just using somebody else's T cells and infuse it to the patient. You save a lot of time up front. You don't need to take the T cells out or engineer them, and T cells are not that tired from seeing all that chemotherapy or treatments in the past. So it sounds really attractive, and there are clinical trials that are doing it. There are also issues and concerns with it, right? What if those T cells attack the patient's body and kind of cause something like graft versus host disease that we see with other diseases? And what if those T cells don't work as good, then we have to. So there are a lot of tweaking that is happening in clinical trials, but it's definitely the future. I think we will see that happening, not in near future, but there are a number of clinical trials that are happening. NK cells are just another type of soldiers in our immune system. So we have done a lot of work on T cells. Technically, you could do all that on NK cells, and there are a number of studies that use donor NK cells, somebody else's NK cells. And there's one example in publications, but there are many more, and there are many groups working on it. So the future will definitely involve number of T cells and NK cells, either from patient themselves or from someone else. They're a little bit behind in terms of the pipeline and the timeline. And the first thing we'll get will be an autologous CAR, patient's own T cells that I mentioned, but the others will come. There clinical trials, we have studies, all major institutions have studies with most of these treatments. So and you can use them in sequence. You can go with the CD19 CAR that will be first available, and if that doesn't work, you still have the option of using other ones.
Dr. Shadman, thanks so much for this. There were unbelievably great questions there that we didn't get to. I apologize for that. I'm going to give you just a minute or two if there's any final thoughts that you wanted to share with patients about CAR T therapy, where it fits in CLL now and in the future, or just any message that you think people should hear.
No, I think it was a great discussion. I think the good news is that we are entering an era of just a completely new type of treatment for CLL patients. And it's not just one drug, it's just a modality which will include a number of drugs. So that's great. Number two, we can combine it with some of our very effective pills, and that makes it even more interesting and effective. Number three, take advantage of these CAR T products. Many people who have enjoyed long remissions from CAR T, 5-6 years before they even existed. They still don't exist for CLL, right? So think about taking advantage of something that doesn't exist. So consider clinical trials and talk to people who know about these things early on. There's no commitment in having a conversation, but that helps you have the information you need to make a good judgment and assess your risk and benefit. At every step of your journey, you need to assess okay, what is my option? What are the alternatives? Which one makes more sense? That only happens if you have enough time and you know about it. These are not type of decisions that you can make in 1 or 2 weeks. So give yourself time, and you either do it or not, but at least you make an informed decision. And we are here to help, and CLL Society has great resources, and hopefully this was helpful for our patients, and looking forward to coming back and talk more about other exciting treatments that we have.
Dr. Shadman, thanks so much for this. I certainly learned a lot, especially in terms of sort of the practical management of this. There were great questions we didn't get to. Please send those, and I'm going to share where to send those to you. We're extraordinarily grateful to the support we got from AstraZeneca, from Beijing, Bristol Myers Squibb, Janssen, and Pharmacyclics for making this event possible. I want to thank everyone for joining us today. And just another big thanks to Dr. Mazyar Shadman, who you can see why I was so lucky at Fred Hutch to have him as one of my doctors, for sharing his time and expertise and just kind of practical nuts and bolts of how to do that. Ask the experts at cllsociety.org. I apologize if we didn't get to your questions. We do have this free CAR-T brochure on the website. We have an electronic version, which you can just download and print, or we can actually send you a physical version of it. We have a post-event survey. I can't emphasize that enough. It's critical that you complete that. Your feedback is important. We listen and we make changes based on your feedback. So please help us with that. Join us for our next webinar, which is on COVID-19. It's going to be an extremely interesting, different one, because we have an infectious disease specialist who's looking at the renaissance of convalescent plasma, looking at some new types of vaccines that might be stimulating T cells. It's going to be going over what's happening, and COVID is rapidly changing. And then in October, we're going to talk about something that Dr. Shadman talked about, and he's really raised a lot of questions here, is how do you sequence your therapies? Do you use drugs and combinations, one after another? What comes first? What comes second? How do you plan this out? This is a huge issue, and it's a changing issue. And we're going to be thinking about how does CAR-T fit into that. So please put those webinars on your calendar. They're going to be amazing. And I'll just remind you that we're a nonprofit. CLL Society is invested in your long life, and please invest in our long life at the CLL Society by supporting our work, and there's a link there. So again, thanks to everyone, thanks to our sponsors, but mostly thanks to the patients and caregivers who attended this, who are their own self-advocates. That's how you live long. That's how you make it happen in CLL. Our motto is smart patients get smart care, and I'm sure everyone is leaving this webinar as a smarter patient. Thank you all.